Hi, this is E. David Crawford, Editor-in-Chief of Grand Rounds in Urology. Joining me is Dr. Daniel Petrylak, who is a professor of medical oncology and urology at Yale University. Dan is a leader in prostate cancer and has been on Grand Rounds a number of times and is going to share with us some late-breaking news of an oral presentation he had about a phase 1 trial that is very exciting about interfering with the androgen receptor and degrading it. Dan?
Thanks, David. So appreciate the opportunity to talk about this data. I'm very excited about it. As we know, drugs such as abiraterone and enzalutamide can develop resistance, and although patients with metastatic disease can have dramatic responses to androgen deprivation therapy, these patients eventually do progress, and they progress by several different mechanisms. This includes upregulation of the androgen receptor point mutations, and probably one of the oldest observations in prostate cancer, the development of an autocrine way of secreting testosterone. So one of the ways that we potentially can overcome resistance is downregulation of androgen receptor or these particular variants. And what the ARV-110 drug does is it exploits a natural mechanism of the cell that we use to chew up proteins. So there's something called a proteasome, and proteins that are marked for degradation are ubiquinated, and then are shuttled to the proteasome, and they're cleaved into small peptides. Well, a PROTAC is a molecule, a small molecule that has 3 different parts. It has a warhead that binds to the target protein, has a hinge region, and has an area that will also bind to the ubiquitin ligase. This trimer that forms basically preferentially targets proteins, and then that's shuttled down to the proteasome for degradation. One PROTAC molecule can take out as many as 400 of the target proteins, so it's recycled. So the ARV-110 molecule has been evaluated preclinically, and what it does, it degrades the androgen receptor, and it degrades the most common variants of the androgen receptor, but it does not degrade the L702H or ARV7. And we did a phase 1 trial that escalated this orally bioavailable drug, and when we reached levels that we actually were able to achieve levels that were seen in the laboratory, that we could see degradation in animal models of the androgen receptor. And in the presentation, I show that in one particular patient. But when we reached that 140 milligram level, and in androgen receptor variants that were degradable, we had 2 patients respond out of the 7. And these have, at the time of the analysis, these were still ongoing. One patient had a 30-week response. And when we looked at their androgen receptor mutations, these were the T878 or the H875 variants that are known to be involved in resistance to abiraterone and enzalutamide. In fact, 77% of our patients had either abiraterone or enzalutamide, and 100% of patients had prior abiraterone. For our patients, they had a median of 6 prior treatments. So we're continuing to escalate our dosages, and we're going to find a recommended phase 2 dose, and then we're going to go on to a phase 2 expansion cohort to define activity.
Dan, that's very encouraging and exciting. We know that the androgen receptor is fairly clever and mutates and amplifies and things like that, and that the common presentation now is failing, as you said, abiraterone, enzalutamide, and some of the other third-generation anti-androgens. So it sounds like the toxicity was not significant and it's fairly well tolerated, right?
Actually, I didn't mention the fact that we did see 2 dose-limiting toxicities. For the most part, it was nausea and vomiting. But we had 2 patients who developed grade 3 or grade 4 transaminitis. And concomitantly, these patients were receiving Lipitor or Rasuvalimab. And we found was that the levels of Lipitor were increased by 4 to 7-fold, and that the ARV-110 molecule will actually hit the pump. So rosuvastatin may not be a drug that we want to give concomitantly. We saw no other episodes in other patients who received other lipid-lowering agents. So again, I think that rosuvastatin may be something that's contraindicated with these patients, but otherwise it was well tolerated.
That's really great. So we look forward to hearing more from you as you evolve through the pathway of phase 2 and phase 3 with this drug. It sounds like a unique mechanism of action. Dan, thanks a lot for sharing this with us.
Great, thank you.